Why are there so many different CFTR mutations in cystic fibrosis? Imagine a gene as a detailed instruction manual for building a specific protein. In cystic fibrosis, the CFTR gene is that manual, and it tells the body how to make a protein that controls the flow of chloride and water across cell membranes. Since the CFTR gene was identified in 1989, scientists have found over 2,500 different mutations in this gene. That's a huge number, and it might seem confusing at first. But the reason for this diversity lies in the gene's size and complexity. The CFTR gene is quite large, made up of 1,480 amino acids, and it has 5 key parts called functional domains. These include 2 transmembrane domains that span the cell membrane, 2 nucleotide-binding domains that help regulate activity, and a regulatory domain that controls the protein's function. Mutations can happen anywhere along this gene, affecting different parts of the protein in various ways. Some mutations prevent the protein from being made at all, while others cause the protein to be made but not fold correctly or reach the cell surface. Some mutations make the channel open improperly or reduce its ability to transport chloride ions. Scientists classify these mutations into 5 main groups based on how they disrupt the CFTR protein. The first group, called Class 1, includes mutations that stop the protein from being produced, often because of errors in the genetic code that create premature stop signals. The second group, Class 2, involves mutations like the common delta 508 mutation, which cause the protein to misfold and be degraded before it can reach the cell surface. The third group, Class 3, includes mutations where the protein gets to the surface but doesn't open properly, so chloride can't pass through. The fourth group, Class 4, involves mutations that make the channel less efficient at transporting chloride even when it's open. The last group, Class 5, results in less protein being made overall. Because these mutations affect different parts of the gene and protein, they lead to a wide range of disease severity and responses to treatment. Some mutations are common worldwide, while others are rare or specific to certain populations. This genetic diversity explains why cystic fibrosis can look different from person to person. When doctors perform genetic testing, knowing exactly which mutations a person carries helps predict how severe the disease might be, guides treatment options, and informs family planning decisions. For example, some newer therapies called CFTR modulators are designed to target specific mutation types, making personalized medicine possible. In summary, the large number of CFTR mutations exists because of the gene's size, its complex structure, and the many ways mutations can interfere with its function. This diversity accounts for the wide variation in symptoms and disease seen in cystic fibrosis. It also highlights the importance of detailed genetic testing and counseling to tailor care to each individual's unique genetic makeup. Always remember, if you or a loved one are dealing with cystic fibrosis, consulting healthcare professionals is essential for accurate diagnosis and personalized treatment planning.